All right. So we're going to talk more about the tree of life now, and a tiny bit of wrap up, although I'm going to skip through a few slides because I was a little bit behind in the other section. I got a little bit further in your section. In general, I know this is, some people have been going to the other section. In general, I'm a little bit better, I think, in the second of the talk, so you're probably better off coming to the second one. And I've corrected a few mistakes here and there and haven't always told people. All right, so what we're going to do today is talk a little bit more about phylogenetic inference and then about some philosophy of the tree of life and then some details about the tree of life. A little hint for everyone. On Monday, very early in the morning our time, the Nobel Prize in Medicine and Physiology will be announced. And then later in the week, they'll do chemistry and physics and literature and economics. In the last few years, at least one, if not all, of the medicine, chemistry, and physics prizes relate in some way to the stuff we're talking about in the class. There will be a bonus question, at least on the midterm, that relates to the Nobel Prizes. So you should pay attention to what they are. And we will, if they really do relate to the class, we will talk about it a little bit. 2 years ago, one of the women who won the Nobel Prize in Medicine grew up in Davis. And she's actually giving a talk in a week or two here. She's coming back to give a talk. All right, so just want to sort of reiterate some things from the previous lecture, and I'm going to skip through a few slides because I already covered this for you here. So I talked about the issue of purines and pyrimidines and transitions and transversions, with transversions being less common, and therefore you want to account for that somehow in phylogenetics. Like I said, you don't have to understand the details of the mechanisms. I've actually worked a lot on this. I'd be happy if people are interested to talk to people about this, but we're not going to expect you to understand these details of the mechanisms. And I talked already about this issue of overcounting some of the changes if they are rarer changes, and that allows us to weight those changes and then come up with a tree that more sort of accurately reflects the reality of evolution. So, because everything blurs together. I went through this in the previous section, right? Earlier today? Okay, good. So the main issue here is that events that are rarer, we want to score, give more points to, because points are bad for a tree. And if your evolutionary tree requires you to hypothesize many rare events occurring, such as the change from a 3-chambered to a 4-chambered heart, or in this case, a transversion, you want to count those more. And the exact way we count them is sometimes complicated. So we can actually use real data to figure out what that score should be. I showed you here, you can give transversions twice the score of transitions. You can give them 3 times the score of transitions. We can actually analyze actual data to figure out what the different rates of those events is. And actually one of the authors of your book, David Hillis, has done a lot of work in this area. He simulated in the lab evolution of viruses, knew exactly the branching order of those viruses because he manipulated them, and then he sequenced genes from those viruses, compared them to each other, and he could calculate the rate of different types of change events in those viruses. And he could even test different phylogenetic reconstruction methods by taking the data and saying, I know the answer. I know what the tree is. Which method works best to figure out the true tree? Which weighting of transitions versus transversions works best to figure out the true tree? So we talked about this with transitions and transversions. It turns out any way you think about it for sort of evolution, there are variations between different lineages or within different parts of the genome of organisms that we might want to account for. Another one that is really important that the parsimony method that we talked about doesn't really deal with is the parsimony method that we dealt with treats changes on all branches equally. So if we have this tree with A, B, C, and D, if there is a change on this branch, I know we did the unrooted, but it's equivalent here with the rooted trees. If there's a change on this branch leading up to D, we consider that the same as if there is a change on the branch leading up to A. But in this rooted tree, the amount of time that this branch represents is higher than the amount of time that this branch represents. A and B share a common ancestor here. The branch that D branched off on separated out longer ago. So there's been more time for changes to occur. We should expect more changes to occur on that branch. We shouldn't count changes equally. So if I was comparing two trees, one tree that had a particular pattern versus this tree, for this tree, if this tree says that a lot of changes occurred on the branch leading up to D, that's pretty reasonable. If for the data that we were looking at, this tree said all the changes occurred on the branch leading up to A, and there were no changes anywhere else in the tree, that's not that likely. This is a short amount of time, and these other branches represent a lot of total time. So you also want to account for different rates of evolution in different lineages when you use, sorry, not different rates of evolution, different amounts of time that the branches represent when we do these methods. Does that basically make sense? That this D branch represents more time, yeah.
So basically you're saying that like the longer the lines on the tree, the more time it represents?
Not yet. So what I'm saying here is that this is a vertical tree.
Yeah.
Here's a root, so I'm drawing a rooted tree. This axis represents time.
Yeah.
And that means that this point here occurred a longer time ago than this point.
Okay.
And that means that this branch going up to D represents more time than the branch from this node to A.
Okay, got it.
And since it represents more time, we should expect to see more changes there. And the parsimony method that we use does not account for this. We treated a change on each branch as being equal. But we should expect more changes on this branch. I'll come to how we deal with this in a minute. But I'm just trying to point out that we didn't deal with this. We just treated changes on every branch as being equal. Another related issue is that even when organisms share a common ancestor with each other, the rate of evolution of organisms is not always uniform. So for example, HIV, the virus that causes AIDS, has an incredibly high mutation rate. This is one of the reasons it's very hard to deal with in terms of drug therapy and vaccine design, etc. Other viruses actually have a relatively low mutation rate. In the time since HIV shared a common ancestor with some other viruses, HIV has evolved an enormous amount. Its rate of evolution per unit time is incredibly high. We should expect more changes to occur on the branch leading up to HIV, as well as all of the branches among the different strains of HIV, than on branches leading up to viruses that evolve more slowly. So we can represent this in evolutionary trees by drawing this type of tree on the right here. So the trees that we've been using in the class are what are called cladograms. For a cladogram, we're just representing the branching orders among organisms. That the term clade just represents this grouping of organisms. A cladogram, like the one on the left, just represents branch orders. This tree on the right instead represents rates of evolution. So if we look here, A and B share a common ancestor here in the tree. I've drawn a longer branch for B. That means that the rate of evolution for B has been higher. The branch length corresponds to the amount of change that has occurred in that organism. A longer branch length means a higher rate of change. Did them?
Yeah,
Mike, kick out, or I think we lost battery. Hang on a second. You have to. You know how to do the battery? There's one here. Maybe. That work? Yeah. Right. So what I'm trying to do here is represent, in essence, something that like HIV, with the branch leading up to B being like HIV, and maybe the branch leading up to A being some other virus. Yeah?
Is there anything significant about the spacing at like the node between A and B and from this?
You mean like this?
A and C? Yeah, like this.
So, yeah, that's a really good question. On this type of tree, no. The only thing that's important on this tree, there are two things. The order of branching, and then the length of the branch. I could have pivoted the branch in any other way. Generally, you try and draw them at 90-degree angles or 45-degree angles, depending on how many things you're drawing. You try and keep the angles. Does that make sense? That angle doesn't mean anything here. So again, if organisms evolve at different rates, and we're just counting each branch as being equal, we're not taking into account the realities of evolution when we try and figure out which tree is the best tree. These trees where you represent evolution in terms of the amount of change are called phylograms, as opposed to the cladograms, which I said before are the ones that just represent the branching pattern. So when we build trees, we probably want to count the ones that occur, the changes that occur in lineages that evolve really rapidly. We should just expect many of those changes. Give them a low score. And anything that occurs on a branch that evolves slowly, give it a high score. Parsimony methods are not very well set up for dealing with this. There are many of these assumptions that I went through. Again, I talked about all types of characters being treated equally, the types of changes being treated equally, like transitions versus transversions, the changes on all branches. I didn't talk about this one, and you don't have to know it, but there are many other examples of assumptions that we're making. In essence, all of these assumptions argue for a slightly different approach to building evolutionary trees. Evolution is much more complex than the simple parsimony method that we showed you accounts for. The parsimony method is really useful for thinking about character evolution, which is why we use it here. But in general, you can account for these variations that I've talked about by using this third class of methods that we've mentioned called likelihood methods. So what likelihood methods do is very similar in concept to parsimony methods. You compare different trees to each other with a particular data set, and you score those trees. But instead of counting the number of changes required for each tree, you calculate a probability, in essence, of generating the data you observe for a particular tree. And the way you do this is you create a model that accounts for time or amount of change, but let's just say time. So there's a rate of particular events. And for every type of change that you're looking at in your data, you assign a rate at which that type of change occurs. And so now when you have a rate parameter, you can account for different lengths of branches. The longer branch that I showed you before leading up to D would have a higher probability of any change occurring on that branch because we're measuring evolution as a rate. So if the rate of evolution is one event per million years and that branch represents 200 million years, you can figure out the likelihood of having a particular change. We can account for the differences in probabilities of transitions and transversions. by giving them different rates. We can say that transversions occur 3 times per every 10 sites per million years. And transitions occur at a different rate. So we build this model of how the characters change. You can use this for any type of data. It works really well for DNA data. You build a model for the types of changes. It becomes very complicated when you try and figure this out because not only are you trying to compare different trees to each other, you're also trying to compare a single tree with different lengths of the branches to each other. So you're trying to simultaneously score trees that have different branching patterns and different lengths on the branches. And you try and calculate which tree has the highest probability of generating the data you observe given a particular model of how sequences change if you're looking at DNA. Does that basically make sense what likelihood methods are doing? We are not going to go into the math of likelihood methods here. I'd butcher it. Brian Moore, who's going to talk later, is actually one of the experts in doing this. And I didn't want to embarrass myself in front of him. But in addition, we're not sort of expecting you to understand the math, but you are expected to understand the principle of how likelihood methods can account for different probabilities of events. Everybody okay with that? Okay, so now we're going to shift topics. We're done mostly with phylogenetic methods, and we're going to actually talk about evolution of real things. And the first thing we're going to do is talk about whether or not there's a single tree of life or multiple trees of life relating organisms on the planet. I don't know, has anybody seen this artwork on campus? Wow, no one. If you go, oh yes, so some have. If you go over to the Oak Grove, near the White Flower gazebo, there's this really nice tree of life sculpture that happens to be on the outhouse over there. I don't know if that's a message or something, but it's pretty cool. They've made this sort of stone sculpture of the tree of life. They've mostly left out microbes, but we'll forgive them for that. There's lots of evolutionary biologists who talk about the tree of life without ever talking about what we're going to do here. My blog is actually called The Tree of Life. Unfortunately now, when people search for it, they find the movie The Tree of Life instead of my blog. I will survive that. But in general, what I'm about to talk to you about is frequently not covered in evolution courses, but it's biodiversity or tree of life related topics. But it's really important to go through this because what we're going to talk about is why people think that everything is related on, in essence, a single tree. Or why do we say a tree of life instead of the trees of life? And so if we drew a tree that related all organisms on the planet, if we put them together onto a single tree, that's what we're going to call the tree of life. It doesn't matter what we name the taxa or anything like that, but if we can place everything on the planet, all organisms, onto this single diagram, we're going to call that generally the tree of life. That implies that all organisms on the planet share a common ancestry if we put them together onto a single tree of life. That is, somewhere at the base of that tree, there is a root node that all organisms are linked to that root node. That's their MRCA, their most recent common ancestor. That's what MRCA stands for. But why can't we have this? Why can't we take all the organisms on the planet and put them on 2 trees that are not connected to each other? And each of those groups might have a common ancestor with each other, but they aren't linked to each other. Does anybody know why? Yeah.
It's just like really improbable.
Why do you think it's really improbable?
Because life has to come from non-life. Like, I don't know, it calls for like all these different conditions and things like that. It's just really improbable.
Yeah, so the argument here is that the probability of, in essence, booting up life from non-living systems is so low that you can't imagine that this happened twice as opposed to once. So that is certainly one of the arguments commonly used. It's what Darwin talked about in a bit in The Origin of Species. Any other reasons why? Yeah. Yeah, so all life uses the same base codes. That is, in general, an argument about the similarity of all life using some similar system, linking together life. And what we're going to talk about is some sort of subtle details of that argument for why we have to think really carefully about what that means. And I just want to go through this because it's really important to understand this aspect. But I am going to talk about exactly what you're saying there in sort of more detail. So what I want to do is basically distinguish between this tree and those trees. Again, this is sort of just theory of evolution here. So the reason that we think that there's one tree is basically if you compare all life to each other, and I actually hadn't thought to talk about this other argument, but that's a really good argument that maybe we can talk about separately. The probability of just life evolving is pretty low, so you argue that all life must have come from a common origin. But what I'm going to talk about is this issue of comparing all organisms to each other, we can find traits that they all share. They're all similar to each other in many features. Characters that are similar or the same across all organisms are called universal traits. The presence of something could be that particular trait. So the presence of everybody using the same genetic code. What does that imply about patterns of common ancestry? If we have a single tree and everything has similarity across that tree, so presence of a common feature, we could infer that the ancestor of all those organisms had that feature, right? If there's no, everybody has the same character state, we can infer that the ancestor had that character state. If we had two trees, and all the organisms have this one particular character state, we can certainly infer that their common ancestor in each of these two branches had that character state. How can we distinguish between these two possibilities? The main issue comes to, related to your probability of life originating, is really a question of, for this particular trait, do we think that it could have evolved twice? Or do we think that it had to have evolved once? So this really is an issue of theory of traits that we observe, whether or not we think that the probability of that trait evolving is either high, and then we could imagine it evolving separately in two different origins of life, or is low, and then we argue that it only evolved once. And this issue is basically an issue of convergent evolution versus homology. So for some traits, let's say the use of water in organisms. All organisms on the planet use water. It's a universal trait. Seems perfectly plausible that if life evolved separately in some place, it might also use water. So the use of water is really not something we can use to distinguish between this pattern and this pattern. Seems perfectly plausible that it could have evolved twice. What people have asked about is, are there any features that we think are so unusual? Life could be one of them. Again, I didn't think of that as a feature, but any features that are so complicated or unlikely to have evolved separately in different branches. And these are what are called universal homologies, as opposed to universal traits. These are features that people have observed in different organisms that appear to be complicated enough or improbable enough that people argue that they are homologous across all organisms. And if they're homologous across all organisms, that implies a single common ancestry of everything. And I'm just going to show you quickly what this looks like on an evolutionary tree. So let's imagine one of those that people have argued, which is the use of DNA as a genetic material. Or even more specifically, the use of A, C, T, and G bases as the bases in DNA. If we imagine this happened twice, we would have to propose two separate origins of the use of DNA and A, C, T, and G as the genetic material. Is that reasonable? This is what you have to ask. Is it reasonable to have had A, C, T, and G evolve separately in two different origins of life? I think it's probably unlikely, but there may be chemical reasons why you might think that it could have happened multiple times. However, there are some things inside organisms that it seems completely unreasonable to argue that they have evolved multiple times in separate lineages. One of them is the ribosome. So the ribosome is this machine that all organisms on the planet use to translate RNA into protein. The machine that all organisms use is very similar across all forms of life. It's made up of two major subunits called a large and a small subunit. Each of those subunits are made up of a complex of proteins. Within each of those protein complexes are individual proteins that are similar across all of life. Each of the parts of the ribosome also has an RNA molecule called ribosomal RNA. And those RNAs are also similar across all of life. So similar to each other that people, myself included, would argue that there's just no way that two separate origins of life would have evolved these ribosomes to be so similar to each other. The same argument has been made about the genetic code. So there's this three-letter genetic code, the particular arrangement of which triplet codes for which amino acid is basically identical across all organisms. And people, again, myself included, would argue that the probability that that has separately evolved in different lineages is so low that we have to link together all of life. So here's the ribosome. It's this complicated structure that translates RNA into proteins. Is it reasonable to hypothesize that this evolved twice? Seems unreasonable. There are many examples of these complicated systems that seem too complicated to have evolved separately. In essence, the central dogma that we use DNA as the genetic material, we transcribe that into RNA and then translate that into proteins. Not only do all organisms do that, but they use very similar complexes to carry out each of those processes. Again, it seems unlikely that that could have happened multiple times. The genetic code is another one. As I've already shown, the ribosome. And here I have a list of many of these so-called universal homologies that are shared across all of life that people have used to argue for a single common ancestry of all of life. Again, there is no theoretical reason why life could not have originated multiple times. We would, however, expect there to be some differences in the core machinery of life between organisms if that happened. And we just don't see those. People basically understand the point here. I know I'm sort of ad nauseum going through it, but point is really important. Universal homologies, features that are complex and homologous across all of life, are what we use to say that there's a single common origin of life. And by saying that, we link together any different groups of things on the tree into a single tree. And this is what Darwin argued in The Origin of Species, argued for why everything should in essence be on a single tree. A single origin of the ribosome, a single origin of the genetic code, a single origin of all of these features. We have to have a single tree of life. So first one question, what about other branching patterns? Does the branching pattern among organisms matter here? It doesn't matter at all. So if I take this tree and infer the common ancestry of all organisms, sharing DNA implies a single origin of life, it doesn't matter what the branching pattern is in the tree. It could look like this. We still would infer a single origin of life. It's the existence of these universal homologies that implies a single origin of life. And this then sets the stage, in essence, for studying the tree of life. Again, perfectly reasonable that there could be multiple separate trees. We don't think that that's the case. So we try and figure out what the branching patterns are among the organisms in the tree of life. So a little thought question before we do that. Can universal homologies be used to infer the branching patterns among organisms in the tree? Why not? Why is it false? Someone over here said no. So anybody want to explain why that's false? There's no variation, exactly. So the presence of a universal homology is what links all of life together, but it doesn't allow us to figure out the relationships among organisms. In essence, the pattern is like the patterns we showed in the data matrix before, where everything was identical. Can't figure out the branching order when everything is identical. Sorry, I'm just going to skip over. We need variation. And this brings us to the last topic for today, which is we need variation, and this turns out to be a big challenge. If we compare organisms to each other, what are we going to use that is shared across all organisms but has variation among them? For many years, people did not know what to do to link together everything on a tree of life, to study this, find something that had variation but was similar across all organisms. So they would compare the diversity of organisms across the tree of life and then draw a tree, but in many cases this was not based upon evidence. So there's a very famous tree by Ernst Haeckel that showed 3 major lineages of life on his tree of life: animals, plants, and what are called protists. That's another term for single-celled eukaryotes, organisms with a nucleus that spend most of their life as single cells. And then at the bottom of his tree, he dumped everything else. And in this tree, there's no real, this is a polytomy coming out showing plants and protists and animals. It's because he had no idea how to link these organisms to each other. This is sort of just a theoretical tree based upon mostly thought exercises. There's another very famous tree you may have seen called the 5-kingdom tree, with plants, animals, and then a new group compared to Haeckel's tree called fungi, which we will hear a lot about in the class. And then protists were lumped together at the bottom of this tree with mostly polytomies. And then organisms without a nucleus, the so-called prokaryotes, were put even further at the base of the tree with no clue as to what to do with them. And the reason for this was that Whittaker had no features that he could use to compare all organisms to each other, where there was variation but characters. In essence, these trees did not do a very good job with microbes. You could compare animals to each other because there were some features that people could use to compare, say, vertebrates. You could line up the skeletons of different organisms. Each bone was slightly different between the different organisms, so there was variation in homologous traits. The homologous trait could be femurs. But if you want to compare organisms that are more and more distantly related, it starts to become hard to find those homologous traits. This generally was the state of affairs until the mid-1970s. And what happened in the mid-1970s mostly came about because of one person, a guy named Carl Woese, who said, let's wait a second. These universal homologies that we can study, the ribosome, for example, the presence of the ribosome is conserved across all organisms. But the exact code that is used in organisms to make the ribosome, the bases in the DNA for the genes that code for the subunits of the ribosome, those vary between organisms. And we can use the sequence of those letters as our information to build a tree. We can create a data matrix that is the order of bases in the gene that codes for a particular part of the ribosome. And we can compare them across all organisms, because those traits are homologous to each other. What he did was he focused on one component of the ribosome called the ribosomal RNA. These are the RNA molecules that make up the ribosome. So somewhere inside this three-dimensional structure are RNA molecules. These RNA molecules actually fold in a complicated way that is not that important for what we're talking about. He ignored that actually when he did this analysis. But what he did was he compared different ribosomal RNA molecules to each other across organisms, read the sequence of those ribosomal RNA molecules, and compared the string of letters in those ribosomal RNAs across different organisms. He made a data matrix that we call an alignment, in essence, where you line up the different molecules to each other. This is just like the data matrix that we analyzed to build the trees with those 9 characters. And then he could compare them to each other. This is analogous to comparing bones between vertebrates. What he was doing was basically lining up the equivalent of femurs in different organisms and looking at the variation within femurs. In his case, the variation in the sequence of letters in the ribosomal RNAs to infer relationships among organisms. It turns out he used the third class of evolutionary reconstruction methods that we haven't talked a lot about, called distance methods. He took the sequences of these ribosomal RNAs and compared them to each other and calculated for each pair the similarity of organism 1 versus organism 2. He then created what's called a distance matrix. So he had Saccharomyces, that's yeast. He compared it to all of these other organisms and created this distance matrix, the difference in their ribosomal RNA sequences. He then took this distance matrix and used methods that we are not going to expect you to know exactly how they work, but they basically group organisms by their similarity to each other. If you do this, you find 3 groups of organisms based upon their ribosomal RNA sequence. If you take that data and build an evolutionary tree where you try and have the tree, the branch lengths in the tree represent the similarity of these ribosomal RNAs, you get a tree that looks like this, where there's one group of organisms here that are the eukaryotes. That is the organisms where the DNA is packaged into a nucleus. And then 2 distinct groups here. One that corresponds to most of the organisms that you are familiar with that do not have a nucleus. These are the bacteria. And then a third group, a third group that was previously unknown of organisms that do not have a nucleus, but are distantly related from the so-called normal bacteria, what he called then the eubacteria. This third group he called the archaebacteria, because many of the organisms at the time in that group had features that he thought were related to features that might have been present in the early Earth, also called the Archean Earth. These are so-called extremophiles, organisms that live in very extreme conditions. So what he had found was a third branch on the tree of life that no one knew anything about prior to comparing the sequences of these ribosomal RNAs to each other. And he also, for the first time, showed that you could place all organisms on the planet onto a tree. Not a tree that you just sort of made up by thought exercises, which is kind of what the previous trees were, but a tree based upon data. And now what he found was this third branch in the tree. Now this tree I'm showing you here is an unrooted tree. Does anybody know why he drew an unrooted tree for this data?
Maybe it might be because he didn't really know like the main common ancestor of all of these organisms.
Right, so maybe he didn't know the common ancestor of all these organisms. That is true. So earlier today, we rooted our unrooted tree how? How did we root our unrooted tree?
Found an outgroup.
Right, so we found an outgroup with some other piece of information. We took the mantid and said the mantid was the outgroup. So here on this tree are all organisms on the planet. What's the outgroup? Life on Mars? There is no outgroup. If your in-group is all organisms on the planet, there is no outgroup that you can use to root this tree. So this is why Woese and other people frequently draw trees of life without a root. They draw them as unrooted trees. I'm just going to skip ahead for a second to show you. Here's a newer version of the tree of life based upon newer data since Carl Woese drew his tree, but it's still drawn as an unrooted tree. And again, because there's no outgroup to all of life. Now, it turns out that in the book and in the class and in many scientific studies, people use a rooted tree of life that looks like this, with 3 branches, the same 3 branches that Woese found, eukaryotes, archaea, and bacteria. This rooted tree is based upon a trick that some people figured out in the late 1980s. So even though there is no outgroup to all organisms on the planet, it turns out if you can find something that may have happened on the branch leading up to the common ancestor of everything, the root branch leading up to the common ancestor of all organisms on the planet, you can hypothesize a rooting to the tree of life. We're going to talk about this a tiny bit on Monday. Not going to expect you to understand exactly how this worked, but the trick that these people came up with relates to duplicated genes, where the gene duplication event, so inside the genome of all organisms are genes. And occasionally within an organism, you can get a gene that makes a copy of itself. It gets duplicated. This is what has happened in the history of our hemoglobin proteins. So for example, many of you have probably heard of alpha globin versus beta globin. There are different forms of the globin proteins that were duplicated sometime long ago in the ancestry of vertebrates. A long, long time ago, in the branch leading up to the common ancestor of all organisms, there were some duplication events that occurred. And again, we're going to just very briefly touch on this on Monday. Analysis of those duplication events is what led to this rooting of the tree of life, where archaea, this third branch in the tree that no one knew about prior to Woese's work, are thought to be the sister group to eukaryotes, to the exclusion of bacteria. So in some of the slides that I skipped over here, I will cover this again on Monday. What we're going to do is talk about the three main branches on the tree: the bacteria, the archaea, and the eukaryotes. We're going to talk about some of the features that they have in common, as well as some of the features that appear to be consistent with this rooting of the tree that may link archaea to eukaryotes. And we're also going to talk about some of their unique features. So what we're going to do on Monday is explore this rooted tree of life and talk about it in more detail, each of these three branches. So that's it.
So, sort of, well, not on the topic
Yeah.
question. It's about the textbook. For Bis002, I had a completely different textbook, and Bis002, we didn't. Really
You didn't
need-
use
we
this
didn't-
textbook for 2B?
We didn't even need that pure explicitly. So it was different. It was
Yeah.
different. It's Bis002 Gerhardt. She has her own book.
Yeah, that's
As you
not
know.
something I support.
Yeah.
But
Right. And so it's kind of weird. So for 2C, I don't know, should I obtain one just for this class?
there are
Should-
going, you could
Because-
use the one in the library. There are going to be parts.
Uh-huh.
So far, everything is you need to know is in lecture.
Mhm.
There are going to be parts of the class where we
I
are
see.
not going to cover some things, and we will tell you that you need to read the book for
I see.
those topics. For my parts, that's going to be a small amount of material.
Mhm.
I don't know about for
Others.
Brian and Brad, how much they're not going to cover in lecture and how much you're going to be required to come from the book. For my parts, I have a couple of specific sections that I will introduce and then say, you need to read, it's like 4 pages. These pages to cover that material. For much of the stuff that I'm doing, the reading will help provide context if you don't know it already. So I'll cover features of bacteria, features of archaea, features of eukaryotes, some of the differences within those groups. In theory, everything I cover in class will be what you mainly need to know. But some of it I will cover fast. Some of it I won't provide the full context and the book will help with. But there are only a couple of parts that I won't cover at all that I will say you need to read this part of the book. That'll be some stuff on the differences between eukaryotic and prokaryotic cells, stuff on lateral gene transfer between different organisms when they steal DNA from each other, and a few of the things on the major groups of bacteria, archaea, and eukaryotes.
Mm-hmm. Okay, I see.
Uh,
For that, would you recommend for that? For this explicitly?
Um,
I have no idea. I was
If
just
you want to save money, you can probably, if you go to the library, I'm sure you can look at the book. I don't know. Almost all of the questions I will ask on the midterm and on the final will be based upon something directly covered in class.
I see.
There will be a few things that will relate to those topics
Right.
that you won't get at all in class. But it'll be a small portion of material. And there will be a few things many things where the reading will make it make more sense.
Would you say maybe there's like an older edition of the book which is really cheap that I could get? Maybe,
You could almost certainly use the 8th edition, which is probably much cheaper
I see.
for my material.
Right.
They did not change much of the material that I'm covering in
Right,
between the
okay.
8th and the 9th.
Okay. Okay. Thank you, Professor.
You're welcome to borrow mine, too.
Oh, really? Okay.
I don't even know where mine is anymore. I'm sure I have the 9th edition somewhere. I have access to the online version, so I don't really look at the.
Oh, okay. Well, that'd be great if you found it.
You probably can check. You should check. You might be able to just check one out from the library.
Okay.
They were supposed to have about 15 copies.
Okay. Okay. Because I don't really want, just for that, just
Yeah.
spend a lot of money. Okay.
I completely understand.
Yeah.
When we designed the course, Gerhardt was supposed to use the book,
What's up with
and
that?
2B.
Just different professors just using different things.
I shouldn't
Yes.
make any comment about that. That was not what they were supposed to do.
Well, I'm assuming she did that because she had some under her name, some stuff in the book. I don't know.
I don't know. I think
don't know.
I've never been kept in the loop on why they decided to do that. I think she felt that the textbook that we're using was